knew how to dress them.
Good afternoon, or midday, I should say. Hopefully everybody's grabbed some snacks and refreshments from the back. Welcome to the Heart Center Research Lecture, monthly research lecture. And we have the privilege today of having one of our own. Dr. Irfan Kathiriya is here to enlighten us on some of the work that he's doing in the lab on the development of congenital heart disease and genomics and transcription. We are extremely lucky to have Irfan with us. He's been a member of UCSF now, coming up on 12 years?
Yes.
Whew. So he did his MD degree and his Ph.D. in the Big D, Dallas, UT Southwestern, right? And then came here for clinical training in pediatrics and anesthesia, and then eventually pediatric cardiac anesthesia. But he's been doing his science throughout that time, and he is tremendous in the anesthesia realm from a clinical standpoint. We're very lucky to have him, and he's going to tell us today how tremendous he is in the lab space. Irfan, thank you.
Thanks, Phil, for that kind introduction. Can you guys hear me through the mic? Okay, great. I just want to put in a quick plug for this new Intramural Catalyst Award for faculty. We're hoping that we can try and promote collaborations, new pilot projects, and we really hope that you'll consider applying. There's a bunch more information online. There's tips on writing a good specific aims page. The application is actually pretty limited, but a lot of the components can then be turned around for NIH grants and other extramural funding. So really it's an honor and a privilege to be here today to be able to share some of our progress in the lab. And so today what I'm going to talk about is heart development and congenital heart disease. And really the approach that we're using is we're trying to combine some of the advantages of using mouse models in vivo, some of the strengths of different platforms, particularly in mouse, so that we can be able to appreciate three-dimensional structure. And then in addition, as a complementary sort of approach, we're also using human iPS cells, so trying to understand how human genes are turned on and off in the context of human cardiomyocytes. And all of this is done in the context of genomics. And genomics, for those of you who may know, genomics is really looking at the context of how genes are turned on and off throughout the entire cell. Thank you. Throughout the cell, so looking at all genes and how they're affected in the context of any perturbation. So just as a brief outline, first I'm going to talk about really our rationale for studying ventricular septal defects, and then I'm going to present really three projects, or progress with three projects. The first is identifying these novel cells that we believe contribute to the interventricular septum and how they're regulated and how they're misregulated to cause ventricular septal defects. And then the second part of the data that I'm going to show is really in the context of human disease, studying human cells, so targeting human cells and then differentiating them to cardiomyocytes and then perturbing them and looking at how changing the dosage of one particular gene, a transcription factor, affects the rest of the genes. And then the third component is really something that I added recently that we've been working on. It's really to try to understand not just how one gene, where you make mutations in one gene, causes congenital heart disease, but how, more commonly, how do genetic modifiers, more commonly to disease, affect congenital heart disease. And so really our hope is by studying this biology that we can gather insights to better help with diagnosis or therapies. So as is well known to this audience, congenital heart disease impacts childhood in terms of morbidity and mortality. And ventricular septal defects are common, both isolated and among complex congenital heart disease. These can be hemodynamically significant, causing heart failure or pulmonary overcirculation, leading to pulmonary hypertension. And it's thought that ventricular septal defects are caused by abnormal development in utero of the interventricular septum. Now, the interventricular septum is important physiologically and biologically because the septation helps to separate both deoxygenated and oxygenated blood, as shown here in this cartoon. And so for those that are just the initiated of what we're talking about here is the ventricular septal defect. So here's the left ventricle and the right ventricle. Here's the interventricular septum. There shouldn't be a connection, at least postnatally, and so this is the ventricular septal defect. And so most causes are really unknown. There's some implications for environment and also genetics. So we're really trying to understand what causes congenital heart disease, specifically ventricular septal defects. And so today, what I'm going to show you are three projects that are interrelated to try to address that. So here is some developmental biology. This is a really simplified view of developmental biology, but I think it's important in terms of the next several slides. So very early on, if you remember from developmental biology, gastrulation is where you have three major germ layers: ectoderm, endoderm, and mesoderm. And the heart is derived from the mesoderm. So at gastrulation, you have mesoderm, primitive streak, that then contributes to the cardiac mesoderm. Early in the cardiac mesoderm, there's at least two distinct heart fields, the first heart field and the second heart field. Here, the first heart field is shaded in green. Subsequently in this talk, I'm going to refer to them as left ventricular progenitors. And then the second heart field, which is just abutting the first heart field in red, I'm going to, again, call them right ventricular progenitors. And the reason I say that is because the green area contributes to the left ventricle and the atria, while the second heart field contributes really to the right ventricle and the outflow tract. So even early, before there's any semblance of a functional heart, these fields are predestined and patterned to end up to specific regions of the heart. And so here's a summary of really a cartoon of the first part of my talk, in which what we believe is we've found some cardiac progenitors. These are marked by transcription factors, MEF2C and TBX5. And each of these transcription factors early on in the gastrulation stage, these cells contribute to the right ventricle and the outflow tract, similar to the second heart field, which I showed earlier. Then you have TBX5 progenitors, which later contribute to the left ventricle and the atria, as shown here in the schematic. And that these two regions are generally mutually exclusive, but there is this small overlap of the two domains, which we consider here are double positive, TBX5 and MEF2, and these end up at the interventricular septum. And so this is the population that I'm going to show you data for today. So this is the basis of that cartoon, the data here. So what we've done, this work here is really a collaboration with another postdoc in the lab, Patrick Devine, who's a pathologist and now a molecular pathology fellow here at UC. And he made a bunch of genetic tools to really try to identify these progenitors and to follow where these descendants of these progenitors ended up in the heart. And so here we're using genetic tools where we can mark cells early in development using a drug. I don't think the details are necessarily that important, but we also have these reporters, so GFP and RFP, green fluorescent protein and red fluorescent protein, where we can simultaneously look at where these descendants end up in the heart. So here, as you can see, we're trying to mark TBX5 positive cells early in development in gastrulation, and we see that those TBX5 positive cells end up in the left ventricle and the atria. So here we consider this the left ventricular lineage. And there's another gene, the MEF2C, specifically the anterior heart field enhancer, where if we mark it early, we see it really contribute to the right ventricle. And when we overlay these two colors, it's hard to appreciate, but there is yellow here, and this is this segment that is this overlap. So I'm going to go ahead and show you a different sort of image using a different reporter that really highlights that. Please, if anybody has any questions, please go ahead and raise your hand. Let's try to make this a little bit less formal. So here in this reporter, what we're trying to do is to only detect those double positive cells. And so here you can see that there are double positive cells. These red cells, they start to create a ring. Now, the time points that I'm showing you, again, this is mouse development. What we're doing is we're highlighting the context of when we believe, when it's been shown morphologically, where the ventricular septum starts to form at embryonic day 10.5, and by embryonic day 14.5, or mid-gestation, it's already complete. So here what I'm showing you are the time points at which ventricular septation is happening in the mouse embryo. And you can see that you have this band-like formation of these double positive cells right at this interventricular groove. And it becomes more refined, and then it occupies this area which is the interventricular septum. We wanted to see what happens much earlier on, because these cardiac progenitors, they're really the genes are really being expressed much earlier in development, not later in development. We wanted to see where the cells ended up, but we also wanted to see where they started out. And so here what we're doing is we're taking really early time points of mouse cardiac development. And embryonic day 7 3/4 is really the beginning of cardiac mesoderm, where you have the two heart fields. And to our surprise, we noticed that these red cells were scattered through this domain of both first heart field and second heart field, not as a sharp boundary between first and heart field. So this salt and pepper pattern was really very unexpected. And this salt and pepper pattern continues during the linear heart tube, even as it progresses, and then somehow it fine-tunes into that band-like formation that happens at 9.5 and then later in 10.5. And we've tried to figure out what the mechanism is. We don't think it's proliferation of the other cells or some cell death. So we're still trying to figure out how these cells are sorted from a mixed pattern to really a very band-like state. So here, so far, what I've shown you is kind of is really trying to characterize these cardiac progenitors and where they end up. They end up in the interventricular septum, and we wanted to know whether or not these cells were even important for biology. And so here, what we did is we used a specific sort of mouse genetic technique where we're misexpressing diphtheria toxin only in the context of the cells that express TBX5 and MEF2C. So again, what we're doing is we're activating the diphtheria toxin in the context really early in development, and we're seeing what happens to the heart later in development. And so you can see it here, the top images here grossly, and then by histology you can see the red cells. And you can very nicely see the right ventricle and the left ventricle. But in the context of when you misexpress diphtheria toxin, the heart looks abnormal. So the right ventricle looks hypoplastic, and the interventricular septum looks pretty disorganized. Now, the one caveat here is that the interventricular septum hasn't finished developing, so we can't say if there's a ventricular septal defect or not. But what we can say is that the interventricular septum is pretty abnormal. So here, I think the take-home points are that the cells that we've identified, the ones that contribute to the interventricular septum, they are necessary for normal biology of the interventricular septum. And then unexpectedly as well, that these cells somehow send signals to the right ventricle, and we're still trying to figure out what that mechanism is, so that the interventricular septum is also somehow contributing to how the ventricular chambers are formed. So now that I've shown you the identity of these cells and I've shown you the function of these cells, we wanted to take a different approach. Thus far, we've used molecular genetics in mice, and so what's called an orthogonal approach is to really see if the genes that we believe should be on early, we wanted to test whether we could detect whether these genes were on early in a different way. And so the way we did that here is to use something, we wanted to look at gene expression, and the current analysis would be by RNA-seq, or where you do high-throughput sequencing of RNA. And because this tissue is so heterogeneous, we really kind of embrace this new technology called single-cell RNA-seq, and that allows us to really see various differences among all the different cells. And so here, in this first set of data, I'm going to show you really the early time points at gastrulation and then at cardiac mesoderm. So here what I'm showing you, each column, so this is RNA-seq data, looking at approximately 150 different cells. So each column represents an individual cell, and each row represents an individual gene. So here I'm showing you about 700 different genes, and we're looking at those genes that are most differentially expressed across these various time points. 2 time points, early and late in gastrulation, and then in the cardiac crescent. And kind of the take-home picture is that in general, the early stages look pretty similar, although they're not identical. And then by cardiac mesoderm, a bunch of genes that are turned on early are turned off, and that there are some genes that are turned on. So we're really concentrating on these genes. So these are, a lot of these genes here are genes that would really turn on the cardiac gene program. And we also wanted to see specifically whether or not the 2 genes that we were most interested in, MEF2C and TBX5, because they mark this unique population, whether or not they were expressed. And really the rationale was because other conventional measures of looking at RNA via other techniques like FISH, we weren't able to detect these genes that early. So really, what I'm showing you here is really the first evidence in which we can see that these 2 genes, even very early on during gastrulation, are turned on, and they get more, and they continue to persist, which is consistent with what we see genetically. So in addition, we also, what I've just shown you is the early time points to look at the cardiac progenitors. And now what I want to do is I want to look at what happens to the descendants in terms of when they occupy the heart at later stages. So here's a similar sort of map. This represents about 200 genes, and again about 150 cells. Each cell that's so we were able to take advantage of all the different reporters, so we could see which one should be a right ventricular lineage cell and which one could be a left ventricular lineage cell and which should be a double positive cell. And that's all marked here below, so that you can see that this group here is really the predominantly double positive lineage. And looking at this sort of it looks like this sort of rug sort of diagram. You can see that here this area is different than this area, so that there are genes here that are turned on in the double positive that are less so in the other areas. And some of these genes include TBX5, which marks the lineage, but then gets restricted later to just this double positive lineage, as well as other transcription factors, TBX3 and islet1. And I'll talk a little bit later in more detail about TBX5. But because TBX5 came out in this sort of really in this sort of analysis, we wanted to see what would happen if we reduced the level of TBX5 to see whether TBX5 regulates these cells. So up to this point, we've identified the cells, we know they're functionally relevant, and now we're trying to figure out how they're regulated. So here what I'm showing you is the context of marking the TBX5 lineage early on in development. So in green is the TBX5 lineage. Red is the double positive lineage, so we can simultaneously look at the double positive lineage and the single lineage. And this figure here is a 3D reconstruction using light sheet microscopy of just the TBX5 lineage. And what's striking by histology at 2 dimensions or even at 3 dimensions is there seems to be this very sharp border, or what we think might be a compartment, where the TBX5 lineage really stops at the midpoint of the interventricular septum. This is pretty unique. There aren't very many areas in biology where this observation is made. And you can see that that also is true for the double positive cells. So we reduce the levels of TBX5 in the context of the TBX5 lineage, and here what we're doing is reducing the level of TBX5 really early in development, so really at gastrulation. And when we look at these hearts, we see that the hearts have a ventricular septal defect. And then when we look more closely by histology, we can see that the nice arrangement that we see of the TBX5 lineage and of the double positive lineage, it's much more disorganized in the context of the mutant interventricular septum. And so our interpretation from this is that that TBX5 early on regulates this lineage. And I think the other important finding from this is that up until this point, we thought that TBX5 had an important role for heart development throughout development. But what this shows is that we've been able to isolate really very early on in development its role later in terms of how it forms the interventricular septum. And data that I'm not going to show you just because of time, we've also been able to isolate really the domain of where TBX5 is expressed, and that's necessary. And we see that really it's only in the context of the overlap with the MEF2C domain.
Irfan, do you have a sense of how much of the septum, how much of the cells of the septum have both markers versus?
Yeah, so we've been able to quantify that. So both markers are about anywhere from 1% to 3%. And so it's really a very small number. Yeah, and that's in the context of the whole heart. And that's relevant because the studies were really difficult because we were only isolating 1% to 3% of an entire heart. So we had some technical hurdles that we had to overcome to be able to get those results. Okay, so for my first part, what I've told you is first we've identified these cells that contribute to the interventricular septum. These cells appear to be important for how the interventricular septum forms, and then also there's this unexpected role of how it influences the other chambers. And then finally that it looks like the TBX5 regulates this double positive population. And so what we're doing now is we think we may have identified some targets that are downstream of TBX5 only in this region, and so we're just following up to see if really that's true. And we're hoping to be able to also get insights as to how, in a developmental biology context, does this border form to really put a marker down that this is where the interventricular septum should be. Okay, so I talked about TBX5. TBX5 is a transcription factor. It is able to bind DNA and is able to turn on genes, and then recently was found to be able to turn off genes. And heterozygous mutations in TBX5 cause an autosomal dominant disease, a really rare disease known as Holt-Oram syndrome, which is a heart-hand syndrome. I remember learning about it in medical school. So what this does is it causes upper limb defects. Shown here, the prolongation of thumbs, or it can be in extremes, it can cause phocomelia. And then in terms of congenital heart disease, it can cause septal defects, atrial septal defects, and ventricular septal defects, as well as conduction abnormalities. And so this example here is really an example of where reducing the level, really just one copy of a gene, is sufficient to cause disease. So this is in a broad category of genes called haploinsufficiency, and that haploinsufficiency causes disease. And so we wanted to know really how does reducing the dosage of this specific transcription factor cause disease. And I should say, you know, Holt-Oram syndrome is really rare, but what we're doing here is we're using this, studying this rare disease to get an entry, really, to get an entry into understanding more broadly mechanisms that contribute to congenital heart disease. And to do this, we used human iPS cells. So we've tried this many different ways. Many of you have heard you can harvest patient cells, skin fibroblasts, and then convert them into human-induced pluripotent stem cells. And those are pluripotent stem cells that then you can coax to go into different lineages. In this context, what we've done is we've used something called genome engineering or gene editing. There are a couple of different ways that you can do it. In the press, there's been a lot of momentum of technology called the CRISPR-Cas9 technology. And basically, these are proteins that lower organisms, such as bacteria, use in self-defense against viruses. And so investigators, Jennifer Doudna at Berkeley, Dr. Charpentier, and then Dr. Zhang at Harvard have really, what they've done is they've really tried to use these proteins to the advantage of biologists. And really the beautiful thing about this is that there are several components to this system. You have a CRISPR-Cas9, which is a protein, which really recognizes this strand. It's called the guide RNA. And really what the guide RNA does is that it creates a information. It's like mailing an envelope to your neighbor. It creates a mailing address, and the guide RNA finds the corresponding DNA, and then with that, the Cas9 can then selectively cut in that location. And so what this has done in the last, this is a finding that probably, I think it was first published 3 or 4 years ago, but has really revolutionized how we can begin to selectively change the genome in this context for disease modeling. So here what I'm going to show you for those that are really interested is just confirmation that we've been able to specifically target TBX5. So we have homozygous clones, and these are actually formally they're compound heterozygous because they have so the CRISPRs can cause what's called there are a couple of mechanisms. One mechanism here is it can cause an insertion of a mutation or a deletion of mutation, and it's non-specific, but because the address is very specific, we can see how that affects the protein. And so in this context, we have some alleles that have a T insertion and some alleles that have an 8 base pair deletion. So the net effect is that they cause the TBX5 to be non-functional. So now that we have these targeted human iPS cells, we've generated both the homozygous and the heterozygous in addition to the control line, so that we can now have an allelic series to be able to see what happens when we reduce the level of TBX5. And because we study the heart, there are protocols where we can coax the cells using chemical modulators to go from the different stages in development, from pluripotency to mesoderm, which we talked about, to cardiac progenitors, which is really the cardiac mesoderm stage, and then to beating cardiomyocytes. And so we can go from pluripotent cells to then in a dish where the whole dish can be beating quite robustly. And it's pretty remarkable that this can be observed in as little as 7 to 8 days. So here what I'm showing you is really the phenotypic analysis of these mutations. So as I told you, there's the parental line, there's a control line. This is a line that has seen the CRISPR but didn't have the mutation, and then the heterozygous mutation, and then the homozygous mutation. And what we observed was a phenotype in that the onset of beating was delayed in a dose-dependent manner based on the mutation. So the homozygous was always the worst, and then the heterozygous was less so, and then the two controls. And this was a consistent pattern with the onset of beating, as well as when you look at differentiation efficiency, which is another sort of metric. And you can see that really on average, the homozygous beats, or the efficiency is much less. We also wanted to look at, because mutations in TBX5 cause AV conduction abnormalities, we wanted to see if maybe there was something intrinsic about these cells. So we looked in the context of a dish, and we have these microscopes that can check the beating frequency. And so here in this graph, in the y-axis, you have the beating rate, and then you have the days of differentiation. And you can see that even in the context of the homozygous, they beat the slowest in a dish compared to the other genotypes. And that pattern really more or less stays consistent during the differentiation protocol. So what it looks like based on phenotypic analysis is that these, in the context of the homozygous mutation, that they don't make the cardiomyocytes. There's something abnormal about the cardiomyocytes. We wanted to see in the context of single cells now, electrophysiology. So was the electrophysiologic defect that we saw in a dish, was it because of individual cells, or was it something that was secreted in the media? And so here, with an electrophysiologist at Gladstone, Michael Lai, he isolated individual cardiomyocytes. So that's what you see here are these recordings, electrophysiologic recordings. So here in the context of control, you can see that the beating frequency is faster than the context of the homozygous. You can also see that just with the action potential, you have something called a plateau phase, and in this context, it's also broader. So in all the metrics that we looked at, both frequency, which is just beating frequency, or the context of the rate of rise, the homozygous was always significantly worse than the control, and that the heterozygous was somewhat intermediate, as in the frequency. And then in the context of really the duration, or the plateau phase, the AP duration, either 50% or 90%, again, was always worse in the homozygous. So here, what we've really demonstrated that that these cells are abnormal in the context of a cultured dish. So we wanted to see really how are genes turned on and off in the context of these various genotypes, and we wanted to take advantage of using single cells because we wanted to see the heterogeneity in the context of a dish. So what I'm showing you here is each individual dot represents a different cell. And here the presentation is a little bit different, but essentially what I'm trying to do is I'm reducing thousands and thousands of dimensions into 2 dimensions. And here what I'm showing you in this figure, in both figures really, are about 18,000 cells from 3 different genotypes. And the time point that we're looking at is really at the cusp of where the homozygous have not started to beat yet, but the controls have. So what I've shown you here, what we've highlighted here in the context of the different colors is all the different genotypes. And the take-home message is that there are a lot of areas where all 3 colors are present. So that means actually those cells, they're the same. But there are some areas, like right here, or right here, or right here, where the genotypes are distinctive. So these are cells where the genes are most similar for just the control cells. These are for the het, and these are for the homozygous. So we wanted to characterize these because this was, at least to our knowledge, is the first time that this had been done. So we wanted to look at really what is the context of the number of cardiomyocytes. So here we're using troponin T and see that actually the RNA-seq data is really very concomitant to what we see by FACS, and that's shown here in brown. But you can see that not every cell is brown, so not every cell is a cardiomyocyte, and these are violin plots just demonstrating this in a different way. We also wanted to look at other mesoderm derivatives, so vascular endothelial cells, and so we saw that there's a small population of KDR positive, which we believe are vascular endothelial cells. We wanted to see if there were any new cell types that, that might appear in the context of the loss of TBX5. And we noticed that there was this one population up here that seemed to be more predominant with the homozygous cells. And so we looked at the genes that were in this population, and we noticed that they were endoderm. They expressed endoderm markers. Now, this is really bizarre because, as I told you early on, the heart is derived from the mesoderm. So why is endoderm appearing in the context of the complete loss of TBX5? It doesn't appear in the heterozygous, and it doesn't appear in the control. And so really we're trying to figure out, does this event happen really early when we'd expect the cells to go through mesoderm and then somehow they deviate to endoderm? Or are these cells that are confused as mesoderm and also express endodermal markers? And then finally what we wanted to do was start to compare different populations to see how, so that we could really get at this issue about dose dependence. And so here what I'm showing you is the relationship of all the different cells versus each other, and down here is a bar graph that shows really the relative amounts of the different genotypes. And what we noticed was there was a population really here, cluster 15, which is predominantly control cells, cluster 14, which is related to it, which is predominantly heterozygous cells, and then again cluster 3, which is predominantly homozygous cells. And so those three clusters shared a bunch of common markers. They were all cardiomyocytes. And so here what I'm showing you is our comparison. So the way these graphs, these are called violin plots, the way they're organized is this is control, these are hets, and these are homozygous. And as you can see that all three populations express cardiac troponin T or TNNT2, a cardiac marker, so these are all cardiomyocytes. We look to see how this system relates to the in vivo mouse system. So in mouse, NPPA is known as a known target of TBX5, and we see that in the context of this in vitro model, this human in vitro model, that NPPA is also downregulated. We found new genes that also seem to be downregulated, like FHL2, and then there's also genes that are also upregulated. And this is a stepwise manner such that it goes based on the dosage. And what's interesting about RSpondin3 is it's a Wnt agonist. So at this time in cardiac development, the Wnt pathway shouldn't be on. So what's weird is that in the context of really of the homozygous mutant, it's somehow, the cells are somehow turning on this factor to turn on a pathway that shouldn't be on. And then finally, we also noticed that there seemed to be a discrepancy between the concept of mouse versus human in context of HAN1, where in mouse, HAN1 should be downregulated, but in this in vitro model in human, it's upregulated. So we're trying to figure out why there's a discordance. It could just be a species difference, or it could be really a technical issue about what we're comparing. Okay, so here what I've shown is that we've been able to generate an in vitro model to study human mutations of TBX5 in the context of cardiomyocytes, and that we see really a dose-dependent phenotype, whether it be beating onset or differentiation efficiency or electrophysiology, and that we think that TBX5 must be playing some role because endoderm is apparent in the context of the loss of TBX5, that maybe that TBX5 is somehow suppressing the onset of endodermal cells, and that that we're now really we're beginning to play with this data to see to really evaluate the dose-dependent changes of gene expression in the context of individual cells in this system. And so follow-up would be to figure out, well, what's causing the abnormal physiology, the electrophysiology, and the phenotype, and then to see if we can confirm this using other methods. Okay, so with the remaining time, I want to just talk very briefly about genetic modifiers. So up to this point, I've talked about identifying these cells that contribute to the interventricular septum. They're marked by TBX5. They're regulated by TBX5. And then I've shown you really a new system to study how human TBX5 affects human cardiomyocytes. And TBX5, as I mentioned, mutations cause this rare syndrome, Holt-Oram syndrome. But really in the context of most diseases are not caused by a single mutation of a single gene. Most, really there's this variability where depending on what your genetic background might make you susceptible or more vulnerable or less vulnerable depending on your genetic background. And so this was also observed in the context of patients with Holt-Oram syndrome. There's variability of their phenotype, which is kind of diagrammed here. I've been focusing on ventricular septal defects, but there are other sorts of abnormalities, really a wide variety. And then also in terms of the prevalence, how penetrant is the phenotype. And so here I kind of stumbled upon this while working with the mouse models for TBX5, trying to understand TBX5 in the context of congenital heart disease in vivo. And in vivo, Benoit Bernot, when he was a postdoc, he had made this mouse line, and he had noticed that in the context of some genetic strains, the mice wouldn't survive to weaning, and in some other genetic strains, they would. And this really poses a problem when you're trying to maintain a mouse colony where the heterozygous line dies, because you can't maintain a mouse colony if you lose the mice with the genetic deletion. And so I was maintaining these lines in the context of a different strain. And then I would outcross these lines to black 6, where the phenotype was most severe. And so here I'm showing you a graph of all the samples that I collected for 4 years. I collected 100 samples, and in the black 6 background, I only collected 1 survival. So usually in the context of Mendelian ratio, you expect 50-50, but so 50% would die and 50% would survive. But here, only 1% in the context of a black 6 background survived. And then when you look at the context of the black Swiss background, the black Swiss, it doesn't get you to 50-50, but you still are able to recover some animals. And so here I'm showing you in these 2 years, it's not 50-50, it's about 30% survival, but that's better. That can maintain a colony. So we started doing these experiments where we were back crossing the black Swiss, and then we would outcross the black 6, and we started noticing that we were getting survivors, that there were pups, mouse pups, that were surviving. So here what I'm showing you is a statistic in which we're looking at weaning. So for mouse gestations anywhere from 18 to 19 days, and then they're like teenagers by day 21, and that's when they can start to wean from the mother. And that's when you separate them. And so really in the context of mating a TBX5 heterozygous, a del het, to a black 6, like I showed you, only 1 survived to weaning. But then what we noticed was there was this generation of brothers, of mouse males, where all of a sudden we started, just by chance, we just started recovering more offspring. And you can see actually the variability is quite, it's pretty variable, this penetrance in the phenotype. So we have some animal. So these are all brothers because they were genotyped all at the same time. And so you have one male where there was no offspring that we could recover. We have one male where we had a lot of offspring more offspring that we recovered and one that was the intermediate. So there was really something that was contributing to this intermediate variability. And so we suspected it was a genetic modifier because of the differences in the background. And so to get a genetic background, genetic modifier, we really started doing a pilot genetic screen using mouse animals. And frankly, this was a complete shot in the dark because in the context of developmental biology and studying genetic modifiers in the context of congenital heart disease many people have tried and have not been successful. But the major advantage here was that we had such extremes in phenotype, where we had a 99% penetrance to then a change to 30% penetrance. And so we were optimistic that maybe we might find something. So as a pilot, what I did was we collected a bunch of samples. So here I'm showing you two family trees. These represent mouse family trees. So you have the two founder males crossed to black 6. So this is the first this is the 0 generation, this is the first generation, and this is the second generation. So what we did is we collected the samples from, these are known as N2 and these are known as N3, where we collected 10 animals, 4 of them were dead and 6 of them were alive. And then we weren't sure what to make of it, so we went ahead and we enlisted Jackson Labs' help, where they would use 150 SNPs to help us genotype this. And again, remember, this is the context of just 6 animals versus 4 animals. And so when we did the initial analysis, surprisingly, there was a trend where we actually found that there were 2 loci at chromosome 8 and chromosome 14 that may contribute somehow to survival or lethality. The p-value is 0.9, but again, remember, the context is only in the context of 10 animals. So what we did is we used this to help do a power analysis, and the power analysis suggested that we should try 40 animals. And so what we've actually done is we've collected 70 animals. And so here's the context of all the different family trees. From all the different males, and one of the interesting observations was, so for example, in this family tree, this is the male across the black 6. The more black 6 after every generation, the more severe the phenotype got. So really, it was either the diluting of the black Swiss or the increasing of the black 6, and that's shown here. So for example, at the 4th generation, we don't ever get any survivors. So we've just harvested all these animals, and we've sent it now to do another microarray study. Instead of using 150 SNPs, now we're going to use 143,000 SNPs, and so that we can get much better coverage. And the hope is that we can get statistical significance to narrow it down to a region, not necessarily to a single SNP or a single gene, but to see whether or not we can narrow it down to a region so that we can then follow up studies to see if we can find the genetic loci. Okay, so we have a few minutes left, but last but not least, lots of people have contributed to this work. I'd especially like to thank my mentor, Benoit Bruneau, who has been extremely supportive in helping me become a better scientist and really supporting all these various projects that are related to TBX5. I mentioned Patrick Devine, who really was the first to identify this double population, and that together we've worked on it to figure out how these cells are regulated. And then lots of other people have worked on these projects. I'd especially like to thank Bayardo, who was my first RA, who's now an MD Ph.D. student at Minnesota, and he really helped push this project forward. And then currently Kavitha and Piyush, who are currently helping me with the current projects. And then last but not least, lots of different funding agencies, past and present, have contributed, have helped support this work, to Benoit and to myself, and without any of that funding support, we couldn't really do any of this. So with that, I'd be happy to take any questions. Thank you.
Can you just, for those of us who don't spend time in labs sequencing things, when you're talking about doing this 143,000, what's the mechanics around it?
So,
How long does that take? What's the cost?
yeah, so actually, it's surprisingly inexpensive to do a SNP array. The SNP array was originally funded by the NIH, and there was a group at University of North Carolina to be able to genotype all various kinds of mouse strains, and so they developed this with a company, with Affymetrix. They created this SNP array, but these days a lot of people don't use SNP arrays because of sequencing costs have come down. But in a relationship, so now they've licensed it to a company. So we've sent the samples to a company, and so for $105 per sample, we can go ahead and scan the 143,000 SNPs, which is awesome because in the context of whole genome sequencing, so whole genome sequencing costs anywhere from $800 to $2,000 depending on which country you have it sequenced, and then whole exome is $400 to $800. So it's significantly less than sequencing. The advantage is that it can be, I can send it out, and 3 weeks later they can send me the data, and then, but that's only the first step. Then we have to analyze it using bioinformatics to figure out, to assign the different roles. Because what I've showed you is a comparison of a heterozygous mutant to heterozygous mutant, and the only difference is whether they survive or die by weaning. As opposed to where you're doing whole genome sequencing, which requires more money, more resources, and a lot more bioinformatics time. But I think eventually, if we can find, the goal is eventually if we can find these loci, then we won't take the 70 samples. We might take a fewer select samples so that we can do whole genome sequencing so we can start to narrow down this region, if we can find it.
Irfan, when you showed that image with the diphtheria toxin and you showed that line, is that line demarcating interventricular septum or the left side of the interventricular septum versus the right side of the interventricular septum?
So the got a lot of slides there. You guys all seeing all my slides. So so here the red. Are you talking about this line, Hytham?
Yeah. And then
So the red represents the double positive. So what we're trying to do is we are expressing the diphtheria toxin in the context of the double positive.
No, but then I think on the next slide you saw that demarcation, maybe a few slides forward.
This one?
Yeah.
Okay.
So are you? I'm just trying to think. Are you saying that the right side of the septum and the left side of the septum have different genetic origins, and then when you say then when you go ahead and say like there's going to be a VSD or thing with this kind of deletion, how does that go into how do we interpret that? And from my standpoint, I'm thinking is the right side and the left side, do they contract differently within the interventricular septum, and are they different cell types?
Yeah, I mean that's a really good question. So I think what our data suggests is that there are at least 3 subpopulations in the context of that of the cardiac mesoderm. So in here, so really what we're looking at is both the second heart field and the first heart field. So there are at least 3 different populations, Hytham, of progenitors at this stage. So you have ones that just contribute to the left ventricle, some that contribute just to the right ventricle, and then some that contribute just at the border in the interventricular septum. Now, the interventricular septum, I guess another way to interpret your question would be that, yes, the left ventricle and the right ventricle both contribute to the interventricular septum. But there is also a population where they both contribute at the center, and that would be at the interventricular septum. And the identity of those cells is different than the identity of the cells that contribute just to the left side of the interventricular septum or just to the right side of the interventricular septum. Does that kind of get at your question? Yeah, so it's a slightly different interpretation.
And can you determine any functional difference between those cells after development? From either a contractile standpoint in your petri dish or in the heart mouse model itself? Their action potential there contract.
Right. So Phil's question is whether or not there's any functional difference between the different cell types, and we have not had, up to this point, we don't have the resolution to look at functionality because there's so few cells, like we talked about. Being able to isolate 1% to 3% for that would be challenging, and then having to, what you'd have to do is to be able to do the electrophysiology, you'd have to replate them. And most of those would die anyways. So we haven't been able to figure out how to do that yet. But a lot of the cells, the interventricular septum cells, they also express a bunch of markers that would contribute to conduction. So it's not clear whether or not these cells are then also the progenitors for the conduction cells, or is it just it's just coincidence. So we just don't know that yet.
Do you have any idea why in your model that had the two deletions, was the right side is the ones that did not form, not the left, right?
For the diphtheria toxin?
Yeah.
So for the diphtheria toxin, I would say that the right is worse than the left. But the left, I don't think the left is necessarily normal, but I think the right has this lumpy, bumpy sort of thing, and it looks hypoplastic more so than the left.
Do you have any explanation or why the right side would be affected?
Why it's more susceptible? So we've had so we wondered if initially it was a technical thing. Based on the mice that we're using, and right now we're pretty confident that it's actually not a technical issue with the different mouse lines that we're using. So the short answer is we don't know yet. Like as I mentioned earlier, we thought maybe that the proliferative rate on the right side might be different than the left, or the level of cell death on the right or the left might be different, but that's not it. So what our current hypothesis is that as this, I think I showed you that the cells are in the salt and pepper pattern, and then they migrate to the center. So our current hypothesis is that as the cells migrate, they secrete some factor from the cells to the cells in the right ventricle and the left ventricle that potentially could turn, give it some information to be normal. And so when we ablate these cells by diphtheria toxin, that function isn't there. So we're trying to pursue that by trying to find if that molecule exists. So we just don't know yet. Ing?
So don't you think that there might be some sort of redundancy of some other factor? Because I feel like when you look at, or at least in humans, if you look at the amount of mutations that come up for the TBX and correlated to the frequency of like these patients that have either hypoplastic RV or septal defects, I don't feel like it's really that high, it's really low, if any. I mean, I don't really see it come up.
Yeah, I mean, it's super, super uncommon. So, so you're right. So TBX5, at least the heterozygous mutation of TBX5 in the context of Holt-Oram syndrome, or even with GWAS studies, it's come up with GWAS, but it's relatively low. I think what I'm trying to illustrate here is to try to study one gene that we think is important based on this rare genetic disease in the context of using that to leverage that information to then learn about other mechanisms. And I would say that from work that I did as a graduate student and then was confirmed later, TBX5 interacts with a lot of different factors that are important to congenital heart disease, GATA proteins and hand proteins and to that effect, which, again, mutations in those single genes cause congenital heart disease, but they're very rare. But I would say that probably what's going on is that there is this core network in the context of a cardiomyocyte during a developing cardiomyocyte that most likely includes some of these players that we've identified, TBX5, GATA4, MEF2, and a bunch of other factors, NKX 2.5. And that there are nuances to the changes in the genetic backgrounds that probably create susceptibility in our patients. That's really hard to prove because you need the numbers. And so that's why I'm using the mouse model to try to get at that. Because I presume those two SNPs, even though the p-value isn't great, those two SNPs land in a region that is not near any gene. It's a gene desert. So statistically, if that ends up panning out, that would suggest that it's not, that there's some genetic loci that acts as a genetic modifier, perhaps because maybe it's an enhancer and it affects the expression of a target gene. And so we just don't know that. I saw a hand over to Walker. Is that okay? That was me encouraging. Oh, Nick.
I did have one question.
Please.
Why that toxin? Did you try other toxins? Did they have the same effect?
Oh, so we haven't tried other toxins. So we tried this toxin because diphtheria toxin is pretty well studied in the context of ablating cells, and it's pretty toxic. And so the advantage of using this system is the mouse genetics is pretty complicated. There are probably at least 6 alleles here that I'm showing you. I've kind of simplified it. But this allele allows us to turn on specifically the diphtheria toxin only where the double positive cells exist. And there weren't really that many other options with that unique sort of, for that unique experimental design. But it's a good question. Great, any other questions? All right, thank you all for your attention.
Well,